Good morning. In 12th class biology, today we have to study Human Genome Project. In this Human Genome Project, we have to study about the genotypic sequence of the haploid set of the gene, which is found in the chromosomes number. In this Human Genome Project, the purpose is to analyze the sequence of genotypes, genes, DNA sequences, polynucleotide sequences in the chromosomal arm, during the formation of chromosomal arm, whatever the genetic sequence have existed, have been sequenced. That is to know, that is to analyze, and its analysis is done in the with the help of the computational devices, so that the analysis can be done with the coordination of the computational tools or devices, so that the major analysis can be done in the welfare of the human disorders, which is the chromosomal disorder, which is the Mendelian disorder. As we have studied the different diseases which is inherited from one generation to another generation, and we have been studying about the hemophilia, sickle cell anemia, Turner syndrome, Down syndromes, and many other diseases, color blindness, so many other diseases which are the hereditary diseases, and which have been studying, and due to this diseases, the many offsprings and many individuals of the new generations are caused, and so to cure this disease, in the search of the sequence of this error DNA sequence, if this error DNA sequence is in capacity to cause the harmful disease to that particular offspring, and that disease is carried throughout the life of that particular person. So if that disease, if that chromosomal arrangement is going to happen in the embryonic condition, if that chromosomal error is going to happen at the fetal condition, in the embryonic condition, in the placental condition, then this Human Genome Project is that sequence which have analyzed to search the error, to search the abnormalities, and it will be purpose to search the cure and the process of cure, how to cure that disease, to that sequence, that inaccurate sequence, so that the offsprings which take birth becomes healthy. But we have to know that in the human genome, 3 into 10 over 9 base pairs are found. And when this Human Genome Project was proposed, the different countries have participated. In 1990, this project was launched, and this was launched by US Department of Energy and National Institute of Health, coordinated by Wellcome Trust of UK and China, Japan, France, Germany, and this project was completed in 2003. And after the completion, it was found that for the analysis of, for sequencing the one base pairs of that particular genome of human, $3 per base pairs were spent. And in this way, the estimate was that an estimated expenditure was 9 billion US dollar. And when this all the sequences, the structural scenario of that base pair, the behavioral aspect of the base pair, and the functional expression of that base pair, genetic expression of that base pair, what the expression, genotypic expression is performed by each and every base pair, that was if written on the book, then we have to imagine if the 1000 letter can be typed in one page of the notebook, then 1000 pages containing copies will be there. If the 1000 pages having in one notebook 1000 pages are there, and 1000 pages, in each and every page 1000 letters are typed, then 3300 such books, notebooks are required to type all the sequences, all the genetic expression, or the structural healthy expression, all the inaccurate genetic expressions will be there. And in this way, that branch of biology in which all these genotypic expressions, and with the help of the computational devices, we are in that condition to sequence all the 3 into 10 over 9 base pairs. That biology is called bioinformatics. So what is the purpose and goal of HGP? Then we have to know that on the 23 chromosomes, in the human genome, it is the goal to identify all the approximately 20,000 to 25,000 genes in the human beings. Second, it is writing that 3 billion chemical base pairs is there, and we have to analyze, and we have to sequence all the 3 billion chemical base pairs of the human DNA. And these all the structural information and the sequences of base pairs have to be uploaded in the database in the computer. And we have to improve the tools for data analysis. We have to improve the tools so that what the computational devices can support the human beings. This computational devices, the tools can help on the basis of analysis of the inaccuracies, and what should be the sequence, like the robotic analytical software should be developed so that it can also, computer can also help us to analyze the sequence, and how to improve the inaccurate DNA sequences in the interest of the human welfare. Last one is that, but all these things are done, but it is to also be we have to also be careful. Yeah, the SGP was also in the guideline of ethical, legal and social issues, so that any human body, yeah, DNA sequences, if it is known, then it should not be doing any other negative works in the as a social scenario, in the legal scenario, in the ethical scenario, so that the human society and civilization and culture can be affected by the scientific ways of experiment or analysis, yeah, by the development of all these means of analysis of the human genome. And before this, the Human Genome Project, other organisms and their genomes also have been sequenced. And in that, yeast is there, bacteria is there, Canaria habitatis elegans, non-pathogenic nematodes are there, Drosophila is there, rice is there. So many organisms are here whose genome has been analyzed, and whose genome has been in the experimental way, how to improve the yield, how to improve the quality, and how to improve the productivity in the highly developing population of the world. Now, in this scenario, we have to also know that this the methodologies means methodologies, yani ki those methods which we have to develop, by which we can know the function of each and every genes. So firstly it is saying expressed sequence tags, and this express sequence tags are those tags in which firstly we have to identify those genes, those DNA segments which can be transcribed in the form of RNA. If that is transcribed, that may be functional, that will be making the protein by the translation, and that will be making the enzymes and will be imparting the physiological and metabolic changes and development works in the body. Second thing is that sequence annotation. If we are not awareing about the what is the coding sequence, what is the non-coding sequence, what is the functional DNA piece, what is the non-functional DNA piece, so we will be using the different restriction nuclease enzymes, endonuclease enzymes, exonuclease enzymes. We will be cutting the whole genome, whole DNA sequences, and making them, and making a proper sequence. It will be not matter there, this will be in the coding and uncoding sequence, but we will be firstly sequencing all the DNA sequence, all the part of the DNA sequences, and later on we will be identifying which one is functional and coding and which one is non-functional and uncoding. After identifying the coding sequences, we know that that coding sequence will be very small. That coding sequence will be very small, whose benefits cannot be obtained, we cannot be gathered. So the second trial is that why not to amplify, why not to make the clones of such coding sequences whose benefits, whose yields, whose expressions can be earned, obtained by the artificial means. So there was the development of BAC and YAC, and these are the bacteria and the yeast, which were prepared for the production of the clones and amplification of the particular DNA sequence whose properties are in the benefits of the human. And next one, we have to know that the DNA sequencers, automated DNA sequencer, yeah, DNA sequences process was developed by the Frederick Sanger, and the Frederick Sanger only not developed the DNA sequences ways, but also sequences also determine the sequencing process of amino acids in the particular protein. And this Human Genome Project was finalized, completed in 2003, and it is written that the sequence of chromosome 1 was completed only in May 2006. And here the 24 chromosomes, 22 autosomes and XY, and the sequence of chromosome 1 was completed in May 2006. It is the line it has been written, and all the chromosomes and whose functions has been uploaded, whose sequences has been done in the computer devices, and it is in the purpose so that the computer, software can also help us in analysis of these DNA sequences and become helpful in making accurate DNA sequences, so that hereditary diseases, so that the improvement of the crops, other plants, so that the productivity can be increased, so that the medicinal plants can be cultivated in the short period of time, so that to fulfill the huge demand of this huge population of the world. So this is the purpose for the Human Genome Project. Thank you.